What an extraordinary operation it must have been to organize this particular meeting and to attract such a extraordinary audience. And also to thank Natalie for a marvelous introduction, which I believe covered very much the waterfront of where CRISPR is at, both scientifically and in relation to the potential ethical issues that are coming. I am a basic scientist, and I am convinced that CRISPR is simple enough that every one of you can leave this room having a very firm grasp of the mechanism that we're talking about. So I've divided my talk into where and when did it arise, what is it, what can it do, where can it take us, and if by chance I have a moment at the end, I want to talk to you about elephants. So, where and when did it arise? The important thing for me is that we did not invent CRISPR. CRISPR arose through the evolution of bacteria, and it arose as a simple and elegant system for them to defend themselves against viral attack. Even bacteria have problems. They have viruses that attack them. But the extraordinary thing is it protects them from reinfection from a virus that they've already experienced before. And in that regard, it is a classic acquired immune system. Now, when I grew up, bacteria were small and pretty simple, and they sure didn't have an acquired immune system. And I will tell you that the first time I read that in the literature, I thought somebody was either very, very inebriated at the time they wrote it, or there's a misprint, because bacteria do not have an acquired immune system. Well, in reality, they do, and it is extremely powerful, and there's been many, many million years to create something which is extraordinarily faithful. So what can it do? I'm going to show you in the first probably third of the talk how it goes about doing what it does. And then there's going to be a slide that says that's the end of CRISPR. And in fact, that would be the end of CRISPR, and in many ways it is the end of CRISPR. The rest of it is possible because of our own tremendous attention to repair mechanisms for our own DNA. So every organism invests a great deal of its energy and its evolutionary history in making sure that its DNA is faithfully reproduced and transmitted from one cell to the next and from one generation to the next. And it is that interface where the power of CRISPR really comes through. And finally, where can it take us? Well, there's been a touch on some of the issues that are arising now as serious things to consider about where it can go. I believe it's a transformative moment. I believe there's been very few things that have arrived in science that have stimulated so much activity because of the extraordinary potential. Whoops, I have to go back. Skipped one. So this is a timeline, and I really want to spend a moment here so that you can appreciate that this is sudden. This is not something that we've lived with for a long time and have had an opportunity to evaluate at many levels, but rather something which has sprung on science so rapidly that even people working closely to the field were not aware of this giant that was running over the hills towards them for some time. I'd like to point out that basic science works in a very, very specific way, and that is people study things because they find them curious, not because they were going to invent CRISPR. So in 1987, I believe, there was a curious sequence found in a bacteria. Curious because it had all of these what are called palindromic repeats. And it was published as, hey, isn't this funny? We haven't seen anything like this before. It was then, sometime later, around 2005, that it was recognized that parts of this funny sequence were in fact not from bacteria, but from foreign DNA. that it somehow managed to be inserted in this curious way. And then, let's see, let's go to 2007. It was the first experimental evidence that there was an adaptive immunity that was mediated through these sequences. And in 2013, it was demonstrated that you could, as an experimentalist, capture some of this mechanism and use it for your own purposes. 2013. And the word CRISPR has now permeated much of our society. This is rather remarkable. And just to give you some idea of what this means to a basic scientist, I don't know if I have the numbers. Yes, I do. In 2007, 10 years ago, when the first experimental evidence for CRISPR immunity was presented, the literature had 20 publications. 20 people or 20 groups had managed to get enough material together to describe something to do with this funny locus that was identified. And there were 20 publications. 10 years later, which is now, there are 6,000 publications, of which 2,000, many more than 2,000, have appeared in the last 12 months. So I hope you appreciate that this is sudden. Remarkably, remarkably fast. All right. So I'm going to use a railroad analogy to exhaustion, and you may not like railroads or trains ever more after this. This is DNA, the typical model of DNA, and you see the double helix with the beautiful base pairing. And I show it because I want you to appreciate the fact that if you have 20 base pairs in a row, you know, AT, GC, et cetera, in a row, that will probably be unique in the genome, that there will be only one place where that particular order of AC or GC or CG will be presented like that. So it's an address, a unique address. Okay? Now, this is to give you an understanding of what CRISPR manages to do. I'm going to ask each one of you to stand at the beginning of this railroad and start walking, and you're going to take 1 second to step between each railroad tie. And you're now going to walk from the beginning of the DNA that you have in a single cell to the end. And now I'm going to ask you how long it will take you to do that. And it will take you 100 years. There are 3.2 billion railroad ties or base pairs in the DNA in one human cell. So it's vast. And what CRISPR can do is not take 100 years to go from one end to the other, but rather to survey the entire thing and within hours or perhaps minutes, identify a specific group of 20 railroad ties out of that 3.2 billion. This is pretty amazing, and for me it's a bit of a shock because I don't really have the capacity to understand things at the molecular level. But this is an analogy that works for me. So it is extraordinarily precise and extraordinarily effective. So this is the heart and soul of CRISPR. Let's see if we can use this. So here is, yes, can you see this little? So we're looking at this piece here. This is RNA. And it has complementary bases that will recognize the same sequence on a DNA. And this thing in the background called Cas9 is a protein that will bind to this particular RNA. And the RNA and the protein in partnership will go find that specific place in that 3.2 billion base pairs. There's 20 base pairs of the RNA that will recognize their complementary base pairs here. And it'll bring the Cas9 to that site. That means it has to survey all the DNA, and it has to reject every place where there's a few base pairs that aren't correct. And when it finds the right one, it has to say, I've arrived, it's going to stop, and it has delivered the Cas9. So What I'd like you to remember about this is this particular image. So here is the guide RNA sniffing along the DNA, and it's going to find the exact 20 base pairs. It's already been sensitized to those. And here is the Cas9. This guy's going to go out there and do something to that site when it gets there. And we're going to talk about his backpack in another part of the talk. But he's going to go down there and find that site. So that's what CRISPR is all about. The boy, the dog, and the backpack. All right? If you can remember that, you've got a pretty good understanding of the CRISPR mechanism. So this is how people talk about it when they're trying to convince basic scientists that they know what they're doing. Here is a virus on the top left-hand side, and it's injecting DNA into this bacteria, this round thing here. And I want you to follow this red thing at the top. The bacteria has a mechanism where it can take a piece of this DNA, it can chop it out, or copy it at least, and introduce it into the bacterial genome at a very specific site. All right?
I'm just going to press the microphone.
Oh, sorry. Is it all right? We can hear? All right. So it takes this piece of DNA from an incoming viral infection, and it inserts it into this unique place in the genome. And that's the original discovery back in 1987, which said, "Gee, this is a funny thing, a funny sequence." And then this thing will make RNA copies that include this copies of this sequence. Now we're going to switch to the yellow one. This came in on an earlier infection from a bacteria and was inserted, or from a virus, and was inserted into the genome. And now it makes this long RNA, which gets chopped up. Here's the yellow one again. The yellow one now associates with this enzyme called CRISPR, Cas9, and it gets carried to its complementary sequence on this incoming DNA from the virus. And typical of how people describe CRISPR-Cas9, the Cas9 function is simply to cut the DNA. Cutting DNA is not healthy for DNA. It falls into pieces. It no longer works. And the bacteria has done its job using this immune system. It has protected itself from reinfection by this virus. Are we okay with this? All right, we're good? Because this is all that evolution delivered in terms of CRISPR-Cas9. There are many, many different versions of the Cas9. There are tremendous biodiversity, which is going to influence the future of the technology quite dramatically. But the net result is that when Cas9 arrives at the site, it is basically able to do one thing, and that's cut the DNA. It either cuts a single strand in some of the new engineered versions, or it cuts both. And that, folks, is the end of the CRISPR story. Not particularly interesting or useful. But in fact, that pretty much is the end of the CRISPR story, because what takes place next is that, as I mentioned, our cells have spent a tremendous amount of energy making sure they can repair DNA. And so if they come across something like this, there is a huge response. And often it is induced by the damage itself to repair it. And so the best I could come up with for an analogy was some arcane type of machine that goes along and fixes railroad tracks, right? This is the DNA repair mechanism that cells invoke to try and repair this. Now, there are two main strategies, and I must say main, because the area of DNA repair, while it is studied intensely, is by far from being completely understood. This may look a little intimidating, but there's really only two strategies of DNA repair that are mentioned here, and it's really quite important that you understand them. So one is called non-homologous end joining. And basically, all that means is the DNA's been cut, and a complex comes along and does whatever it possibly can to stick these ends back together again. And it's horribly sloppy at doing it because its main job is to make sure the DNA is reattached. So it means that you can drop out pieces of DNA, you can even stick in other pieces that happen to be floating around. You create what are called indels for insertions or deletions. So you create mutations at that site. And many human diseases are in fact caused by naturally occurring indels in gene sequences. The other strategy, we're okay with the first? It's just, you cut a rope and basically you tie a knot in it to connect it again. And the knot is not like it was before, it's got a mutation in it, the knot itself. The other strategy is to use homology-directed repair. And this is extraordinarily elegant. It means that a break in the DNA can be repaired by having a template which spans the break and recognizes and is complementary to sequences on either side, such that when homologous directed repair has taken place, you no longer have the break, but you have put something in which is of your design. So if you have a mutation and you want to get rid of it, you simply put in the sequence that has the right sequence and not the mutation. You cut somewhere in the middle of all this. Homology-directed repair exchanges the mutant piece for the repaired one, and you're in good shape. You're a way to go. If you want to put in something big, like even a new gene, you can do that. You can put in very, very large templates. So we're good on that one? These are the two strategies. And remember, this has nothing to do with CRISPR. This has only a response to cut DNA. We managed to cut the DNA with CRISPR, but now we're in the realm of the DNA repair mechanism that cells have. All right, that could also be the end of CRISPR. We cut and we're allowed to do one of two things: tie a knot in the DNA or elegantly repair it or insert something, but not so. This is really a beautiful image of the actual molecular structure of CRISPR-Cas9 associated with DNA. And I show you this not because there's going to be a test on it at the end of the talk, but because it shows the sophistication that was brought to bear on understanding this mechanism immediately once it was understood. And that has meant that we can make versions of Cas9 that now can go to the site, park themselves there, but not cut it. Or cut it only one strand, or not cut it at all. And that raises some extraordinary issues when it comes to the backpack this boy's carrying. So again, we have the dog as the guide RNA taking us to the site. The boy is the Cas9, but this time he doesn't know how to cut DNA. He just knows how to park himself when he gets to the exact address that the experimenter defined beforehand. Taught the dog how to smell for it and set them out to do it. But what's in the backpack now can be anything that you attach to the dead Cas9 protein. And that turns out to be very, very interesting. So we've already heard about the first two packages that get delivered. And those are just the Cas9 itself. And the first is the non-homologous end joining, and the second is the homology-directed repair. And you said you were all good on that, so now we know those two strategies. You cut the DNA and the cell repairs them. But now we're going to use Cas9 that does not have the ability to cut DNA. And look what we can do with it. We're looking at D now. We can attach something that activates a gene. Imagine being able to go into a cell and say, that particular gene I want to be turned on. It could be a gene that's only expressed during embryonic development. It could be a gene that has very beneficial effects. It could be so many things that we can now think about doing. And we can send the boy, the dog, the backpack to that place and turn on that gene. We can put a gene, a switch for off, so we can shut off a gene. And if you want to treat cancer, maybe shutting off an essential gene is a great way to go, for example. The other is in E, we can put effector domains. Probably all of you are well aware of the term epigenetics. That is, changes that take place on DNA that affect its expression, but do not affect the base pair sequence. And we are now in a position to send with this package things to a region of the genome and change the epigenetic modifications on the DNA, which may have profound effects on expression levels, et cetera, or response to other things in the cell. And finally, something that's probably of the least interest to the majority of people worrying about sort of the application of CRISPR is the last one, where we simply tag the Cas9 with a fluorescent marker. But for the first time, it allows basic scientists to go and see where in the nucleus a particular gene is at all stages of the cell cycle. This is the first time we've ever been in a position to actually track what's going on inside a nucleus. Really quite extraordinary. So the next part, you've already heard from Natalie. The CRISPR is applicable to all species, plant, animal, bacteria, of course. You've probably come across, or some of you may have seen clinical trials for retinal diseases, for muscle, stem cell therapy, modifying induced pluripotent stem cells from individuals to repair the defect. And then you have their cells with a mutation that's been fixed. Germline modification has been done now in an enormous number of species. Mushrooms have been on the market, I believe, for over a year now. Corn, rice, maize, rats, pigs, primates, all of these have been subject to CRISPR-Cas9 gene editing that has been taking place in either zygotes or early embryos, so that it's in all cells, including their own gametes, and therefore they can transmit that change in what's called the germline to the next generation. And we've also heard about gene drive. And I wanted to pause on this just for a moment. Natalie already mentioned it because it's quite appropriately one to worry about. It is possible, and I'm not going to go into the mechanism of how, but using CRISPR, it is possible to create a circumstance where a particular copy, we all have two copies of our genes, and often they're slightly different. The differences are called alleles, but you can use CRISPR to create a circumstance where instead of these things segregating into half the progeny gets one allele and half gets the other, 100% of the progeny only get one. And so you can think now about putting in lethal genes so that you can wipe out a population like those that carry malaria or Zika. And you do have to raise the issues about what that means to the ecology. But there are very clever ways of maybe getting around some of these issues. For example, if you edit the gene that makes the receptor that allows the mosquito to be a host for malaria, then you still have the mosquito, but it's no longer capable of being a vector. I mean, it's an extraordinary moment that this type of power is becoming a reality. And just in case you think this is a theoretical, there have already been field trials. So it is really important that people know about it and think about it and raise their concerns. And finally, I won't say anything about biological warfare, except for the fact that my lab, which is me and two other people at the moment, is very, very active in editing the genomes of mice. That's 3 people with very limited equipment. And you can buy the components that you need, the specific CRISPR components, from a repository called Adgene for under $200. There is no threshold that has ever been lower for the weaponizing of biology. That now we have CRISPR on our hands. However, that's all I'm going to say about it. I am going to go on, if I have just a moment, and it'll only be one moment, to describe what I think is the elephant. In one lifetime, and I mean mine, when I was born, the structure of DNA was not known. We didn't know that it was a double helix. We didn't know how it contained the information that cells use to go about organizing what life is. We knew remarkably little about how it was protected, looked after. We finally got to the point where we understood the basic idea of DNA, RNA, protein, which has been modified many, many times. And then somewhere around maybe 30 years ago, we got really serious about trying to read the sequence of DNA. And we became really good at that, and now I think you all know that you can get your own DNA sequenced for maybe a few $1,000s. Quite extraordinary. And there's hundreds and hundreds of species that we know the exact DNA, the exact sequence of those railroad ties, whether they're AT or GC at each particular place. And that has been profound in breeding programs. It has been extremely useful, and it is the foundation of much of modern human genetics, which is trying to understand how our genotype affects our susceptibility to disease, for example. But with CRISPR, we now know how to write DNA. We really have eclipsed evolution, because now we can put into an organism basically any sequence we want, anywhere we want. It is beyond belief to me that from my birth to now, we have gone from no understanding to this extraordinary level of control. And so the elephant to me, well, certainly we must be very concerned about CRISPR. The elephant to me is much more, what's going to happen if I had another lifetime? Or in fact, the young people in this audience, in your lifetime. If that much happened in mine, what's going to happen in yours? And in that context, I see CRISPR as a very powerful tool added to the armamentarium of biologists. But what I see coming is an acceleration of our understanding of biology, which is going to yearly be giving us new opportunities to control what we have. Particularly in my world of neuroscience, one can look forward, I sincerely believe, to a time not too distant in the future where we may have the power to control our circuitry and the behavior of our circuitry in our brains through this type of manipulation in ways that weren't even imaginable when I started out. And on that note, I think I am at the end. The elephant has been revealed. All right.